Thank you, Jama. I appreciate it. And thank you, Gloria and Jama, for inviting me to this wonderful city. And I appreciate the honor of coming here and presenting our experience doing several clinical trials at Duke. So at Duke, we have typically focused on several strategies, which include molecular targeted therapy, convection-enhanced delivery, immunotherapy, and this is a process that's still in the lab, but soon to be clinical reality in terms of oncolytic virus therapy. We also have traditionally had a lot of interest in drug resistance mechanisms at Duke, but I'm not going to talk about this today. This is the schema of our translational lab. Duke has an extremely vibrant translational laboratory scheme, and including Dr. Bigner, who does immunotherapy, intracavitary immunotherapies, Dr. Friedman, who does chemoresistance, John Sampson, Duane Mitchell, who do both convection-enhanced delivery and immunotherapy work, Dr. Matthias Gromier, who does oncolytic poliovirus, and all these kind of feed into our clinical trials program at the Adult Brain Tumor Center. And then some of these ideas trickle down to the pediatric world. And we also interface, we are one of the 8 institutions which is a member of the Pediatric Brain Tumor Consortium. And we also have a relationship with St. Jude and in terms of clinical trials and also tissue transfer. And Har-Yan, who's also a molecular biologist who does a lot of work on digital karyotyping, and one of his papers just came out in the New England Journal regarding isocitrate dehydrogenase in malignant gliomas. This is a broad schema of the pathways that are affected in malignant gliomas. And I'm not going to go into that except to say that these are all the multiple targeted inhibitors that are currently available for these pathways. But what is important to remember is that in an established tumor, there are multiple pathways that are affected. And it's clearly that, as all the other speakers alluded to, one pathway or one inhibitor is not going to do the trick. And we need to be focusing on all these different mechanisms that lead to a tumor and to hopefully achieve a cure. And these are some of the targeted agents that are currently available and are being tested at Duke, including bevacizumab, Tarceva, sorafenib, sunitinib, vatilanib, and Zactima, which is also the agent that Alberto was talking about. And this is just to show you that a single agent therapy rarely works in these tumors. These are all the multiple trials that have been done over the last several years. And these are color-coded depending upon the different pathways that are involved. And as you can see, even though there are some transient responses seen, in the majority of patients does not lead to sustained tumor control. And again, it is possible that we are dealing with an established tumor. And this is just an example to show you that when we look at angiogenesis as a pathway, it differs depending upon the stage of the tumor. A tumor that is early tumor has immature tumor blood vessels and easily responds to a single angiogenesis inhibitor. But as the tumor continues to grow, you can see that the same tumor does not respond to the single agent angiogenesis inhibitor anymore and requires additional therapies. And as you can see here, this is an established tumor, and using 2 agents, one targeting the angiogenesis pathway in terms of the VEGF pathway, and also another agent that targets the PDGFR pathway seems to do the trick here. And that's again going back to this. There's 2 components to the angiogenesis pathway. One is the endothelial cells of the tumor, as well as in the more established tumors, you start to see the pericytes, which are the supporting cells of the endothelium. And in the mature tumors, it's going to be hard to just target one pathway and say, okay, the tumor is going to get better with that. The pericytes also need to be targeted in order to achieve better tumor control. So again, as Mark alluded to, a tumor is not just a simple entity. It is both just tumor cells plus conscripted cells from the body, including not only cancer cells, but immature tumor blood vessels, immune cells, fibroblasts, and other cells. And unless you target all these components of the tumor, it's going to be hard to achieve long-term tumor control. And again, this is just to show you that you need to be able to target not just one pathway, but multiple pathways. And these are some of the schemes that have been reported in the literature in terms of how well we could use our molecular targeted agents in a better way. And this is an example of showing you could either maximize the inhibition of a target, or you could maximize the inhibition of a single pathway, not only starting from the receptor, but downstream as well. Or you could inhibit parallel pathways, or you could also inhibit a pathway that leads to feedback loop augmentation here, in this case, mTOR. In this case, there's a feedback loop inhibition through insulin receptor, insulin growth factors. And an agent, not only mTOR, but an agent that targets the insulin growth factor might achieve a better control of the tumor. And these are some of the, using these particular strategies, these are some of the combination trials that are currently in the clinic. These are some of the combination regimens that we are testing at Duke, including Tarceva plus EGFR inhibitor plus an mTOR inhibitor. Zactima, which is also the VEGF inhibitor plus EGFR inhibitor, along with PDGFR inhibitor, which is Gleevec. Using lapatinib plus pazopanib, which is a compound that has both it's a multi-targeted kinase inhibitor, VEGF, c-KIT, etc. And then, of course, bevacizumab plus an EGFR inhibitor. We also are exploring the use of conventional molecular targeted agents with chemotherapy drugs. And these are some of the examples of combination therapies that we are either testing at Duke or in other centers. Just to show you an example, we had previously shown that Gleevec plus hydroxyurea has shown some benefit in patients with malignant gliomas, both grade 3 and grade 4 tumors. And that is based on the PDGFR inhibition of Gleevec, as well as its other properties of lowering interstitial pressure within the tumor by suppressing the pericyte in the endothelium. And that improves the entry of chemotherapy into the tumor. And also the combination of hydroxyurea, which is a ribonucleotide reductase inhibitor. This combination seems to work in a proportion of patients with malignant gliomas. And we are testing whether the addition of an additional VEGF-R2 inhibitor, this is again a multi-targeted kinase inhibitor, PTK787, to see if we could improve survival in these patients. And this is an example of, we all have one example coming out of these trials. But anyway, this is a 74-year-old recurrent GBM patient who had failed XRT, temozolomide, and followed by temozolomide plus CPT11. And he had progressive disease after 6 cycles. And with this combination, he had this impressive response, and which was sustained for 10 cycles. But a word of caution about combination therapies that added toxicity might or might not lead to synergistic effects, but might definitely add to toxicity, as Alberto clearly showed. And these are some of the trials of using these multi-targeted inhibitors in combination with others. And as you can see, that there is improvement in the response rates in these trials, but you can also see there's a steady increase in the risk of hypertension, proteinuria, thrombocytopenia, and other bad side effects. So it's important to realize that when we combine these agents, we have to be careful about the added toxicity that we bring on. So this is a list of some of the trials that we're currently doing, and these are the Avastin-based trials. After showing some impressive response with Avastin plus CPT11 in recurrent malignant gliomas, we moved on to trying this agent in a newly diagnosed setting. And this is the combination of Avastin plus RT plus temozolomide during radiation in newly diagnosed patients, followed by Avastin plus temozolomide plus CPT11. And this is a phase 1, phase 2 trial. It's accrued 80 plus patients, and the Avastin was well tolerated. I'm not allowed to talk about the data, other than to say that we did see some impressive survival rates in these patients. And this data will be presented at the ASCO meeting this year. We've also seen some preclinical data of efficacy of combining Avastin plus the proteasome inhibitor called Velcade. And based on this preclinical data, we moved it to the adult setting. In the clinical setting in recurrent GBM patients. It's a phase 2 trial which is ongoing at this time. Again, in the mode of combining Avastin with cytotoxic therapies, we have used Avastin plus chemo or etoposide in recurrent patients in a phase 2 setting, and this is ongoing. We're also testing the use of Avastin plus CPT11 following gliadal wafer placement again in the recurrent setting. And this is also ongoing. We're testing other angiogenesis, anti-angiogenesis compounds. One of them is angiocept, which is actually it's one of those new scaffold proteins, which in this case is fibronectin, which is universally present in all tumors and all. And this fibronectin has 3 domains, and one of the domains has been modified to make it an anti-VEGF-R2 compound. And this is called C332. It's a pegylated form of the drug, and it's given in combination with CPT11 in recurrent patients. These are some of the other molecular targeted agents we're using. Pazopanib plus lapatinib, as I already mentioned. This is targeting the EGFR ERB2 pathway along with a multi-targeted kinase which targets VEGF-R2 and other kinases. This is also the recurrent setting. It's a phase 1 trial that is still ongoing. This is a pan-EGFR inhibitor combined with temozolomide, again in a phase 1 setting. We've moved on to use Zactima in combination with Gleevec and Hydrea. And then we have Zactima in combination with chemotherapy, and then again, dasatinib plus erlotinib. I'd like to talk a little bit about the bevacizumab story, both in adults and peds. As you all know, bevacizumab is a recombinant humanized monoclonal antibody that is recognizes all VEGF isoforms, has a half-life of about 17 to 21 days. It decreases both blood flow and permeability in tumors. And its mechanism of action could be related to its decreasing the interstitial pressure within the tumor that facilitates better chemotherapy entry into the tumor. It might also potentially affect the vascular niche of tumor stem cells. And this might be one mechanism where Avastin by itself could be an effective agent. We've seen Duke was one of the first institutions to try Avastin plus CPT11 in adults with recurrent malignant gliomas. And we found an objective response rate of 63%, one of the best response rates we have seen in a recurrent setting, with an impressive prolongation of survival of 6 months progression-free survival of about 38%. This made us move this trial to the pediatric setting, and we started a phase 2 trial in pediatrics through the Pediatric Brain Tumor Consortium using a combination of Avastin plus CPT11. What we did was we gave patients Avastin as a single dose for the first couple of weeks, and then did perfusion imaging before and after. And we also did some additional biology studies, which I'll talk about in a minute. And then afterwards, we started giving these patients CPT11 in combination with Avastin every 2 weeks. Patients could get this treatment for up to 2 years in the absence of unacceptable toxicity or disease progression. We, of course, stratified patients according to their enzyme-inducing anticonvulsant status. Patients who were on enzyme-inducing anticonvulsants obviously got a higher dose of IRINOTECAN. The adults also, after doing the initial phase 2 study, moved on to test the bevacizumab in a randomized fashion. This is a Genentech registration trial where they randomized patients to bevacizumab alone versus bevacizumab plus irinotecan. And in patients who got single-agent bevacizumab who failed were allowed to get irinotecan as salvage therapy. Their primary endpoints were 6-month progression-free survival and radiographic response. And these were the eligibility criteria for that trial. At the same time, NCI also, under the leadership of Howard Fine, did a single-arm phase 2 study of using bevacizumab alone and patients, and a small cohort of those patients who failed bevacizumab got CPT-11 additionally. Just talking about the PBTC-22 trial, we had two strata initially, stratum A and stratum B. Stratum A was high-grade gliomas, and stratum B was diffuse pontine gliomas in recurrence. We had 18 patients in high-grade gliomas and 17 in stratum B. The median age was 16.2 years and 8.8 years. And half of them were high-grade gliomas, grade 4 gliomas, and half of them were grade 3. Only 2 patients were on enzyme-inducing anticonvulsants. As you can see, the median number of courses received in stratum A was 3 with a range of 1 to 8, and stratum B was 2 with a range of 1 to 9. There were 15 evaluable patients in stratum A, and as you can see, in contrast to the adult study, that we had no objective responses. We had 1 patient with stable disease that lasted for 112 plus. In fact, this should be, yes, there was 1 patient with stable disease at the time of presentation of 112 plus days. I was still continuing to get treatment. And in the diffuse pontine glioma study, again, there was no objective responses. There's 1 patient who was getting treatment at 271 plus days. As you can see, the majority of patients had progressive disease. But what we found was, when we looked at the data, that 7 out of these patients, 7 patients with recurrent malignant glioma had sustained stable disease that lasted for more than 12 weeks. And these were anywhere between 86 to up to 548 days. There was 1 patient who also was invaluable because he had a dosing error, but had received 10 courses of treatment with a stable disease that lasted for 287 days. And again, in the brainstem glioma group, there were 5 patients who had sustained stable disease that were lasting from anywhere between 100 days to about 271 days. And out of these patients, 3 patients in stratum A and 4 in stratum B came off protocol not for toxicity, but did not come off for progressive disease, but came off study because of adverse events. And this is an overall a table that shows you the comparison between the adult and the pediatric trials that I alluded to before. This is PBTC-22. And this is for patients in stratum A, which is the high-grade glioma stratum. There were 17 patients. Their median progression-free survival was 4.2 months, and a 6-month progression-free survival was 40.2%. When you look at the adult randomized study, Genentech registration trial of bevacizumab versus bevacizumab plus CPT-11, you see that the median progression-free survival is very similar to ours, even though obviously the numbers are much higher in the adult group, with fairly similar, almost similar 6-month progression-free survival. So you can see here the bevacizumab plus CPT-11 had a slightly higher median progression-free survival, and as well as a 6-month progression-free survival. Well, these were not the analysis were not, it was not planned to compare the 2 arms of the trial. So we cannot say whether one arm was superior to the other or not. In the adult single-agent trial from the NCI, again, the sample size was 48 patients. The median progression-free survival was 4 months, and the 6-month progression-free survival was 29%. And these are significantly higher than the historical controls of patients with recurrent malignant glioma who were treated with salvage therapies. And I think this data is going to be the basis for considering whether VEVACIZUMAB should be approved for patients with recurrent malignant glioma or not. I think it will get approved based on this data. We did some correlative studies, perfusion studies in these patients before and after single-agent bevacizumab, and also as the patients continue to get Avastin CPT-11. This is just, I'm going to show you 1 patient with diffuse pontine glioma who was on treatment for 55 days. And as you can see, this is the flare image of these patients. This is the contrast-enhanced views before and after. And this is the perfusion scan at the time, at the baseline scan, and then during the course of therapy. As you can see here, in the 2-week, or this is about a month interval, you can see that there's a definite decrease in the perfusion. There's also a decrease in the flare, as expected, and also a decrease in the gadolinium enhancement following Avastin. And then as time goes on, it gets better. But then you can see in this perfusion scan, the KPS, the permeability ratio just increases significantly. And then a month later, this patient was found to have progressive disease. So clearly, Avastin is doing what it's expected to do, which is shutting off permeability and also decreasing the peritumoral edema in these tumors. But clearly, it does not seem to be a sustained response. As I mentioned before, we did a biology correlative study where we took peripheral blood mononuclear cells from these patients before and after 2 doses of Avastin. As you all know, peripheral blood mononuclear cells express VEGF-R2 quite robustly. And we wanted to see if the phosphorylation status on the VEGF-R2 was decreased following the administration of bevacizumab. And again, we saw an interesting dichotomy of response in these patients. In 7 patients, we saw that the VEGF-R2 phosphorylation was decreased over a period of time. But again, we saw, we also saw in another 6 patients, we saw that paradoxically the phosphorylation was increased in these patients. So it's hard to understand what was going on in these patients, particularly in this group. Why would the phosphorylation of VEGF-R2 go up in the face of bevacizumab? There might be multiple factors that might be ongoing, but at least this shows you that there is a pharmacodynamic effect of VEGF, of bevacizumab on the receptor. It's almost a surrogate marker. It's not actually on the tumor, but it's fairly close to the tumor on the endothelial cells. So there are multiple resistance mechanisms that have been postulated for in terms of why anti-VEGF therapy does not work. And clearly, tumors are very smart, and they have multiple redundant pathways for angiogenesis. And see, these are some of the factors that have been listed as potential mechanisms of why some of these agents are not working in our patients. Basic fibroblast growth factor has been found to be increased in these patients. The Notch pathway has been found to be active. Then there is an increasing body of evidence that placental growth factor might be an important mechanism for resistance mechanism in patients who get anti-angiogenesis therapies. And also we talked about the increase in the mature blood vessels, wherein you need just not a VEGF receptor inhibitor, but you also need to target the pericytes. Again, we get all this impressive data in the preclinical setting where we're looking at mouse models. And clearly, mouse models are clearly different from the human disease, where human disease is much more complex. The stroma, the human stroma, also plays a big part in how these tumors progress and metastasize. And while preclinical models are important to initially at least test these targets, validate these targets, we have to take those with a pinch of salt. And most of the efficacious mouse model experiments have not really worked in the human setting. Talk a little bit about the cancer stem cell approach. We showed Dr. Jeremy Rich and all at Duke, when he was at Duke, showed that radiation increased the CD133 population in malignant glioma. There was an increasing fraction of cancer stem cells within that resistant population and had a distinct survival advantage after radiation. And when he used a checkpoint kinase inhibitor in these cells, there was immediate abrogation of the resistance, and you can see that these cells became very sensitive. So clearly, the stem cells can be targeted by some of these approaches and made more sensitive to both radiation and cytotoxic chemotherapy. Again, as others have pointed out, that we need to really look at cancer stem cells as an approach to therapy in order to have sustained tumor cell kill or sustained stability of these tumors. And we've also already pointed out VEGF inhibitors could target the vascular niche in these patients. We talked about checkpoint kinase inhibitors that can also suppress the radiation resistance. We currently have a Notch inhibitor phase 1 trial through the Pediatric Brain Tumor Consortium, MK502. And Notch pathway is very active in stem cell populations. We should also look at combination approaches to targeting stem cells. And I'm going to talk a little bit about immunotherapy soon, and there are strategies where you could target these stem cells, cancer stem cells, through immunotherapy. I'm not going to spend much time, this has already been mentioned, especially in the context of diffuse pontine glioma, but convection-enhanced delivery has been a pet project at Duke, and we've initiated several clinical trials using convection-enhanced delivery. Convection-enhanced delivery is a novel strategy that bypasses the blood-brain barrier, and using a pressure gradient is able to infuse large molecules directly into the tumor milieu, in comparison to diffusion, where you get an initial rise but then rapidly falls off. Whereas with convection-enhanced delivery under pressure gradient, you can get a sustained increase in the concentration and distribution of a compound directly into the tumor. And this is particularly so in malignant gliomas, where there's this leading tail of satellite lesions that goes away from the main mass. And these need to be targeted in order to get sustained cure in these patients. And convection-enhanced delivery by this wide distribution around the tumor cavity can achieve that. We worked with this compound called Synergizes vesidotox, which is actually a fusion protein of IL-13 in combination with Pseudomonas exotoxin. As Alberto initially showed you, interleukin-13 is highly expressed in malignant gliomas and is not expressed in normal brain tissue. So the integrin receptor, IL-13 receptor, is expressed on malignant gliomas and not in normal tissue. So we're combining interleukin-13 in combination with Pseudomonas exotoxin, wherein we take out the binding domain of the Pseudomonas exotoxin, but leave the domain 2 and 3, which allows the toxin to get into the cell and inhibit protein synthesis. So this cartoon shows you that in a tumor that has the IL-13 receptor, this compound, this fusion protein, will target the IL-13 receptor, internalize, and cause inhibition of protein synthesis and cell death. In a normal cell that does not have the receptor, this compound is completely inert. So there have been several trials in the adult population with malignant gliomas using 2 or 3 catheters that are strategically placed around the tumor cavity and allowing for the infusion of these agents. And there have been single institution studies that have shown improvement in survival, but a randomized study, an international randomized study of this treatment did not show an improvement in survival as compared to conventional therapy, which is BCNU wafers. But I think convection-enhanced delivery, and part of the problem with that kind of therapy is the placement of catheters. And if the catheter placement is not correct, the treatment is not going to work. So you need expert neurosurgeons who are familiar with the placement of catheters and in order to do these therapies, or a real-time review of the catheter placement before the infusion is done. So I think convection-enhanced delivery still has life, and I think is still being pursued. In terms of brainstem gliomas, as it's been already mentioned, there have been 2 patients infused directly into the pons with IL-13 PU33 38QR, and it's been found to be safe. This is the example of this patient getting this infusion, and this is the distribution that was seen in this patient. There's only transient lethargy and worsening of 6th-nerve palsy in this patient. Otherwise, the patient was completely fine. And actually, there's going to be a phase 1 trial that's going to open soon in the National Cancer Institute, wherein 6 high-grade glioma patients and 6 diffuse pontine glioma patients are going to be treated with convection-enhanced delivery using this compound. I think this trial is still being reviewed, and it's not started yet. So these are the other strategies that could be, other compounds that could be used. We, in fact, are testing EGFR variant 3 immunotoxin by convection-enhanced delivery at Duke. It's a phase 1 study that's ongoing. You can do chemotherapeutic agents directly into the tumor. We're also testing this compound called, this is a derivative of 06-benzoyl-guanine, which is an inhibitor of methylguanine methyltransferase. And we use this compound for convection-enhanced delivery because it's more soluble. And therefore, by directly infusing this compound into the tumor, you're minimizing systemic exposure. And then when you give temozolomide, there's preferential toxicity on the tumor and less toxicity on the body. I'll move on to immunotherapy. And again, immunotherapy is also a big pet project at Duke. And we're using dendritic cell-based vaccine therapy along with adoptive lymphocyte infusions. So again, dendritic cells are antigen-presenting cells, and in the presence of a tumor-specific target can be taught to recognize tumor antigens and to stimulate various immune cells within the body and help to, therefore, control tumor growth. So the targets that we have tested at Duke are EGFR variant 3 and cytomegalovirus. As you all know, EGFR receptor mutations is a deletional, EGFR variant 3 is a deletional mutation of the EGFR gene, resulting in loss of these amino acids. And when it's joined, it creates a new glycine here, making this a very specific target. So we use this particular peptide to immunize patients who express variant 3 in their tumors. And about anywhere between 25 to 60% of patients with adults with malignant glioma express the EGFR variant 3 along with EGFR amplification. And EGFR variant 3 has been found to be an independent prognostic factor for poor survival in adult patients with malignant gliomas. So this is the ACTIVATE trial where patients get leukapheresis up front, but also get radiation plus temozolomide following surgical resection. Then they get randomized to getting this variant 3 peptide along with an adjuvant called KLH and GM-CSF. The other group gets saline plus GM-CSF. This is just for the 3 injections. And then they have immunological monitoring by pheresis. And then all patients then subsequently get monthly injections of this peptide along with GM-CSF. And patients are tested for delayed type hypersensitivity. We are looking at radiological response. And on patients who relapse, we get biopsies to look for antigen escape. So there's a thought that chemotherapy might impair immune responses, but in actual fact, it actually augments immune responses against the tumor. And as you can see in this one case report that's been published recently in Neuro-Oncology from the MD Anderson Cancer Center, using the same strategy, we find that following temozolomide, we actually get an augmentation of humoral responses. And also, we get an increase in the regulatory T cell responses. And what happens is, also with temozolomide, we get a decrease in the T regulatory cells, which is a proportion of the CD4 population that actually suppresses immune responses against tumors. So chemotherapy has a beneficial effect in suppressing the Treg cells and allowing the activating cells titers to increase. And this is just a survival curve that shows you that patients who get the EGFR variant 3 vaccine have a better survival in comparison to the historical controls. And these have been matched controls to make sure that there's no disparity in the clinical presentation and age and other things, the classical prognostic factors that determine survival. This again, the numbers here. The interesting thing that we saw in this group was that in comparison to pre-vaccination, during the post-vaccination recurrence, the variant, although EGFR still continues to be expressed, there was no variant 3 expression in these patients. So clearly, there was antigenic escape in these tumors at the time of progression. At least in terms of suppressing the EGFR variant 3 expressing cells, we were successful. There's currently a randomized study, of
comparing this peptide versus nothing. So patients get conventional treatment versus antigen vaccine therapy. And this trial is ongoing at this point. It's a multi-institutional trial run by Celldex Pharmaceuticals. The CMV story is interesting. CMV is a tumor-associated antigen that's expressed in multiple tumors, multiple solid tumors in adults, including malignant gliomas. And this is a trial that we did at, this is a biology study that we did at Duke, where we found that CMV-specific antigens, especially PP65 and IE1, were robustly expressed in patient glioblastoma tissue by immunohistochemistry. And this was confirmed by in situ hybridization. And this again to show you that the PP65 is expressed in glioblastoma cells, as is the IE1 antigen, and is absent in normal tissue. So based on this, we did a CMV antigen trial where we, again, leukapheresis, patients get RT plus temozolomide, and are randomized to getting an infusion of activated T lymphocytes versus the other group does not get anything. And then all patients get immunized with DC dendritic cells that have been exposed to PP65 in vitro. And then we, again, just like the previous trial, we look for delayed type hypersensitivity and immune responses, as well as radiological responses. Again, this is the scheme of the study. This is the amount of lymphocytes patients get. And these are the parameters that we are looking at. And this is, again, the survival curve. Again, in 13 patients, again, these numbers are small, but there is clearly an improvement in time to progression in these patients as compared to historical controls. So I think immunotherapy is an important strategy that we need to be looking at, and we're actually starting the process of looking at to see if CMV is actually expressed in some of the pediatric tumors, and we're actually looking at immunomeduloblastoma as our model, because there's been some previous studies, especially from Sweden, that has shown that about 80% of medulloblastomas express CMV, and we're actually trying to confirm that in our samples here at Duke. And if that pans out, we might be initiating a trial in recurrent medulloblastomas. But I was also thinking that Alberto's frozen tissue as well as the paraffin tissue might be useful to look at CMV cells expression in diffuse pontine gliomas. And that could lead the way to potentially an immunotherapy approach to diffuse pontine gliomas. Lastly, I would just talk a little bit about this oncolytic poliovirus therapy that we are being tested. We've been testing this in the lab for the last 10, 15 years now through Dr. Matthias Gromier. And it's going to soon hopefully become a clinical reality for adult patients with malignant gliomas, and potentially later on for pediatrics too. So we all know about the poliovirus, and the neurovirulence of the poliovirus is due to its IRES elements, which is actually a region within the 5' untranslated region of the RNA of the virus that allows it to translate protein in the absence of a cap-dependent mechanism. The receptor for poliovirus has been well described. It's the CD155, and it's widely expressed in different tumors, especially malignant gliomas. What we have done is replace the poliovirus IRES with the IRES of a common cold virus that abolishes the neurovirulence of this of the poliovirus. And you might wonder what makes it non-virulent. That is actually, it's been well described, the double-strand ribosomal binding protein and this other NF45, the heterodimer, prevents the cap-independent translation of PVS ribose cells in neuronal cells in the normal tissue, but it's not able to do that in tumor cells. But it's extremely highly tumoricidal. And it's been shown to be extremely safe in rats and cynomolgus monkeys. I'm just again to show you the cap-dependent translation in normal cells and the cap-independent translation in viruses. And this is the structure of the poliovirus, and this is the IRES element in the 5' untranslated region of the virus that's been replaced with the common cold virus. And this is again to show you that there is robust, in malignant glioma cells, there is a high expression of CD155 in these tumor cells. Multiple tumor cell lines and primary tumor tissue expresses CD155, and very little expression in the normal brain and in the spinal cord. And it's extremely tumoricidal in these patients. This is, as you all know, poliovirus causes meningitis, so it seems to be a natural repository for these viruses. So we are looking at testing this PBS ripo in neoplastic meningitis caused by malignant gliomas. And in this, so there was good expression of CD155 in this tumor tissue. And the tumor cells were injected into the rat spinal cord, creating neoplastic meningitis. And then the poliovirus was also injected subsequently, and clearly, in comparison to controls, there was prolongation of survival in the animals that got the inactivated poliovirus. So I think if this strategy works, and this is the current status of this particular recombinant virus, it's being developed by the NCI. It has undergone extensive pre-IND discussion with the FDA and recently entered production for a clinical lot, and for direct toxicology testing in cynomolgus monkeys. And I'm told that all these studies have panned out really well, and there's no toxicity. And we are actually planning, if everything works, we will be conducting an intracavitary injection of poliovirus into these in adult patients with malignant gliomas. And if found to be safe in adults, we could consider, we are actually looking at intrathecal injection as a way of doing these trials in kids with neoplastic meningitis, particularly from medulloblastoma. And recent lab data also suggests that medulloblastoma cells are extremely sensitive to the poliovirus, this recombinant poliovirus. We're also looking at some of the intrinsic molecular pathways that determine the efficacy of this virus in these tumor cells. And we actually have an R01 that's been recently approved for continuing our work in testing this compound in medulloblastoma. So, but using convection-enhanced delivery, if this virus is found to be safe, we could look at potential trials in diffuse spontaneous gliomas as well. I'm going to stop there, and thank you for your attention.